Good evening. Today I'm going to talk to you about anatomy and physiology of muscle cell, the muscle fiber. As you know, the muscles contain muscle bundles, and muscle bundles have muscle cells and muscle fibers. The skeletal muscles are under the voluntary control of the individual, unlike the cardiac muscle and smooth muscles, which not under the control of are not under the control of an individual. They have automaticity and rhythmicity also. And muscles are attached to the bones with a collagenous tissue you call tendons. And basic purpose of skeletal muscle is the movement. Movement. I'm raising my finger, so this is the job of the skeletal muscle. But I have no control on my cardiac muscles. All right? And what I mean to say is the skeletal muscles form 40% of the total body weight. And skeletal muscle has skeletal muscle bundles. They have numerous muscle cells and muscle fibers. And I'm going to talk to you about the microanatomy of the muscle cell and the muscle or the muscle fiber. After I finish this brief talk, I'll show you the sketches which I have made. Muscle cells, muscle fibers are, cylindrical in shape, and they are multinucleated. And the nuclei of muscle cells, they lie just underneath its cell wall, which is called sarcolemma. The wall of the muscle fiber, muscle cell, is called sarcolemma. And this muscle cell, a muscle fiber, has got transverse striations, and it has got thousands of muscle fibrils which are longitudinally stacked. All right. So the muscle fiber, whenever it gets impulse, it contracts. So therefore, as you've seen that there are a large number of muscles fibers may get impulse and contract depending upon the extent and of power, force required to do the job. If I want to pick up a pen, you see that. So just you see, it's a very small amount of number of muscles are involved, muscle fibers are involved, muscle cells are involved, muscles are involved. But if I want to lift heavy object, then of course the lot of muscle fibers must get the impulse to do that kind of a job. So the muscle fibers or the muscle cells, you can say, they have glycogen inside it in the sarcoplasm. And there are other structures also which generally you find in other cells like endoplasmic reticulum, or there's rough sarcoplasmic reticulum also. It has got glycogen and glucose inside its cell in the muscle fiber, and these muscle cells also have certain proteins which are called actin and myosin. What kind of proteins it has? It has got actin and myosin. They are filamentous structures, right? Then we have troponin and tropomyosin also. So these structures, which we call as the part of sarcomere has got the components in it which are called actin and the myosin. They are stacked longitudinally and they glide over each other when these contract, when the impulse comes from the motor nerve fiber. Then these actin and myosin, we'll discuss it later, they contract and glide over each other, and then subsequently they relax also. Now, what happens is that the actins, the actin are thin. They are, within the sarcoplasm, they are, enclosing this, what you call myosin, which is a thicker filament. Myosin is thicker filament, and actin is a thinner filament. And when the contraction occurs, they glide over each other and come to a, then they come to its normal position. Once the muscle impulse is generated, they contract and then they come back to its original position. Now, we have, a system of what you call that, you can say that moment the muscle fiber gets an impulse. So these muscle cells and muscle fibers, or the muscle fibers, they have got, one is resting membrane potential, resting membrane potential, resting membrane potential, which is minus 80 millivolts. And the inside of the muscle cell during resting phase are negatively charged, negatively charged. All right. When a muscle impulse, when the impulse comes to the muscle fibers, what happens is that a impulse is generated and the muscle fiber contracts. This actin and myosin, actin glides over the myosin. What happens during this phase is that we have to understand the peculiar structure of the muscle cell and the nerve fibers. You should understand that muscle fiber will contract only when an impulse is generated. Whenever this impulse is generated, it is transmitted to the muscle fiber. As you know, the motor nerves, they are myelinated, right? And moment they come in contact with the muscle cell, the myelination disappears and the junction, the motor nerve, branches into multiple nerve fibers. Ultimately, they are called multiple nerve terminals. Multiple nerve terminals are there. They are microvesicles in the terminal part of motor nerve fiber, which comes in contact with the muscle fiber. And this contact, the place where the terminal motor nerves, fibers, and the muscle fiber, they come in contact with each other, there is a thickening of the cell wall of the muscle fiber. You see, which is called sarcolemma. And this contact point is called neuromuscular junction. What is it called? Neuromuscular junction. And at that stage, the nerve is without the myelin sheath. Is that clear to you? Once the impulse is generated, the terminal vesicles, microvesicles, they contain acetylcholine. And there is a thickening of the sarcolemma, as I've already told you. There is a neuromuscular junction, and the thickened area is called motor end plate. And this motor end plate is folded. The sarcolemma is folded just because there should be maximum contact between the terminal fibers and the motor end plate. And the motor end plate has got receptors. The moment this impulse comes, what happens? The acetylcholine is released and the receptors in the motor end plate, they pick it up and sodium goes inside and an impulse is generated. Once the impulse is generated, the muscle fiber contract. This is called depolarization, the contraction of skeletal muscle fiber. Once it contracts to form a certain task, so what happens is the action potential, this is called action potential, which was in resting membrane potential. This is 80 millivolts, but now it goes to 20 millivolts. 20 millivolts, positive 20 millivolts, and then it returns back to its resting membrane potential. So therefore, what I mean to say is, when this impulse is transmitted by the motor nerve fibers, motor nerve fibers at the neuromuscular junction, the actin and myosin, the impulse goes to the actin and myosin, which are contained in the sarcomere. Sarcomere, they are longitudinally stacked. Actin on the outside and myosin filament are thicker and inside. So they glide over each other and there is a contraction. Is that right? So this phase of impulse coming to the neuromuscular junction, causing contractions, cause depolarization, and when it comes back to its original position, it's called repolarization. All right. So what is it has some kind of energy is required always to perform a specific task. So therefore, we have, what are the source of energy? When a muscle is moving, if you want to undertake some muscular work, you want to involve your muscles and therefore the muscle fibers. So therefore, you require some energy, and energy is stored in the form of ATP, as you know it very well. ATP is the source of energy for so that the muscle fibers contract. But the ATP is limited, it's got limitation, its number and strength, so it can barely suffice for few of the muscles, smaller muscle actions. So therefore, we have another source of energy, creatine phosphate. So creatine phosphate, it's added up, it joins with the ADP, and ATP is released. Again, the ATP, this creatine phosphate, the store of creatine phosphate is again limited. Creatine phosphate would barely suffice to undertake contractions for around 100 times, 100 contractions. Therefore, its source of energy is again limited. So what the next thing is that you want to undertake much more work from the muscle. So therefore, you require glycogen and glucose, which is available in skeletal muscle in plenty. So this glycogen and glucose is hydrolyzed. So therefore, what happens is it releases the, for the sake of energy, the pyruvic acid, which ultimately gets converted to carbon dioxide and water through the citric acid cycle. And citric acid cycle with its aerobic metabolism will require glucose to generate energy for the muscular contraction. So therefore, ATP is formed, and one molecule of glucose will generate 38 molecules of ATP. Is that clear? So this is the basically it's metabolism. But we'll discuss this cardiac muscle business later on, and I would like to show you through the diagram or a sketch, whatever you call it, you'll probably understand it better, and kindly focus on the sketches which I'm going to show you, in which I'll demonstrate to you the anatomy of the muscle fiber, the sarcomere, the actin and myosin, how they act and where are they placed, and I'll also explain to you the role of neuromuscular junction, and finally this thing, what I've already talked to you, the utilization of glycogen and glucose to generate ATP. Kindly look here. These longitudinal muscle cells which you see here, they are the muscle fibers. You can clearly see their muscle cells are multinucleated. The muscle cell is longitudinally structured, and it has got transverse striations. You can clearly see the transverse striations, all right? See that these are the transverse striations. Okay. And besides that, there are longitudinally placed myofibrils are placed. So this basic structure of muscle fiber is the muscle cell is cylindrical in shape. It is multinucleated. There are several thousands of myofibrils which are longitudinally placed, and it goes striations. The muscle wall is called sarcolemma. As you see it here, the sarcolemma is the cell wall. All right? And coming back to the sarcomere, what does it contain? What the muscle cell contains is, you find that the sarcomeres are longitudinally placed. It has got two components, the protein components. The outside you find is actin filament, and within that you find the myosin filament. You find the actin filament and there is a myosin filament. And the moment the impulse comes through the motor nerve fibers, the sodium goes inside, the muscle fibers contract, and the actin glides over the myosin, and subsequently it relaxes also. This whole structure is called sarcomere, and it also has source of energy which you call the glycogen and the glucose. Now coming next is that you find there is a motor nerve fiber. Motor nerve, the terminal part is branching and splice and provides and conveys, transfers signal, the impulse which is generated to the muscle fibers. And one nerve may be, providing motor nerve fibers, may be giving impulse to few muscle cells or muscle fibers. Now this junction where there is a contact, you find this thickened region is there. This is called neuromuscular junction. You find the thick region. This is called neuromuscular junction. You find it here. This is the neuromuscular junction, right? And there are so many neuromuscular junctions here in this picture. Now this neuromuscular junction has where the nerve fibers come in contact with it. There is a motor end plate, and this neuromuscular junction, this thickened ridge, is folded. Now this is folded. You see if you enlarge the microscopic picture, the neuromuscular junction, the thickened ridge over the sarcolemma is folded. The sarcolemma is folded. There are so many folds in sarcolemma. The reason is you find the axon here, which is part of the nerve fiber. So the terminal nerve fiber and the neuromuscular junction has got better contact, more area is covered by it, right? The purpose of having foldings in the sarcolemma is that motor nerve fiber should come in contact with larger surface area of the neuromuscular junction and the motor end plate. And you find motor end plate, you find there are receptors here. You can see the receptors, right? These are the receptors over this, and these are folds in sarcolemma. You find it like this here. This is the terminal motor nerve fiber, right? And it has got what mitochondria also you can see, and it has got microvesicles. These are the microvesicles. And once the impulse comes to this microvesicles, the terminal buttons, they call it, acetylcholine is released. The acetylcholine comes in contact with the receptors here, and the muscle fiber contracts, and this contraction of muscle fiber is known as depolarization. And then acetylcholine disappears, and thereafter it comes to its resting position, into resting membrane potential. Is that right? So I wanted to show you this thing. Let us read a little bit that muscles are composed of in a microscopic state by so many muscle cells or muscle fibers. It has got a cell wall which is called sarcolemma. It is multinucleated cell. They are transverse striations and it has got longitudinally placed myofibrils. Is that right? And the main source of energy for muscle fiber contraction is glycogen and the glucose. And the contraction comes into play when the impulse comes to the neuromuscular junction where acetylcholine is released, and this impulse goes to within the muscle fiber where actin and the myosin you find, the actin and myosin, the glide over. This is the actin, which is a thin filament outside, and inside is the myosin. They glide over each other and there is a contraction. So that is the thing. And then finally you can say neuromuscular junction is located on the muscle cell wall. This is a thickened ridge here you can find, and if you magnify the structure will be something like that. The nerve fibers, motor nerve fibers are myelinated, but the moment it enters the sarcolemma, the myelination goes, is absent from that side, and terminal buttons of the motor nerve fibers contain microvesicles having acetylcholine, and acetylcholine is a mediator of contraction, which are picked up by the receptors, and therefore sodium enters inside the cell, and this impulse is transmitted to sarcomeres, the actin and the myosin, to effect the muscle contraction. Thank you very much.